Hello there, and welcome back to Sleep Science Time. It's genuinely lovely to have you here again. Thank you for choosing this particular corner of the internet to wind down, to let the day's chaos settle into something quieter, something gentler. Tonight, we're going on rather a different sort of journey. We're not travelling through space or deep time, though there'll be plenty of both along the way. No, tonight we're going somewhere far more personal. We're going to explore the instruction manual that's inside every single one of your cells right now. The code that built you, maintains you, and connects you to every living thing that's ever existed on this planet. We're talking about DNA. It's quite a story, actually. From its origins in some warm ancient pool billions of years ago, through every ancestor you've ever had, right up to the present moment where humans have learned not just to read this code, but to edit it. And then we'll peer forward into a future where we might redesign ourselves entirely. It's a journey that spans 3.5 billion years and somehow ends with you, right here, probably under a blanket, possibly questioning your life choices. Before we start, I'm curious, where are you listening from tonight? Are you tucked up in Manchester at midnight, listening from Melbourne as the sun comes up? Perhaps you're in Montreal, pretending this counts as educational content while you're supposed to be working. Leave a comment and let me know. I always find it fascinating and wonderful about imagining all of you scattered across the globe, carrying the same basic genetic code, just with delightfully different variations. And if you enjoy these gentle explorations into science, a like and a subscription would be tremendously appreciated. It genuinely helps this little channel reach more people who enjoy falling asleep to the sound of someone whittering on about nucleotides. For those of you who've been here before, thank you. Your continued presence in this small community of curious, sleepy minds means more than you know. Now, the usual disclaimer, I'm not trying to keep you awake until dawn. Sleep is very much the goal here. But if you do find yourself still listening by the end, I hope you'll drift off with your mind quietly full of double helixes, evolutionary accidents, and the rather beautiful fact that you're carrying a 3.5 billion year old code that's never once been broken. Not in your entire family line. Not once. Rather remarkable when you think about it. Right then, shall we begin? There's something profoundly strange about DNA that we don't often stop to appreciate. Right now, as you're lying there listening to this, every single cell in your body contains a complete instruction manual for building you. Your eye colour, your height, whether you can roll your tongue, your tendency to sneeze in bright sunlight, all of it, written in a code so small that if you printed it out, it would fill about 130 telephone directories. And yet, for most of human history, we had absolutely no idea this code existed. We knew children resembled their parents, obviously, but the mechanism, the actual physical substance that carried traits from one generation to the next, was completely mysterious. It was hidden in plain sight, coiled up inside every cell, waiting for us to develop the tools to see it. So what actually is DNA? It's a molecule. A very long, very special molecule, but fundamentally just a chemical compound. Think of it as a ladder that's been twisted into a spiral, creating the double helix. The sides are made of sugar and phosphate molecules. The interesting bits are the rungs. Each rung is made of two chemical bases paired together, and here's where it gets elegant. There are only 4 types of bases in DNA, just 4. Scientists called them adenine, thymine, guanine and cytosine, or A, T, G, and C if you prefer not to tie your tongue in knots while falling asleep. That's it. 4 letters. The entire blueprint for building a human being is written using an alphabet of just 4 letters. It's like writing all of Shakespeare's plays using only A, B, C, and D. Except instead of plays, it's eyeballs and immune systems and the ability to feel embarrassment. These 4 bases pair up in a very specific way. A always pairs with T, G always pairs with C. Always. This pairing rule means that if you know the sequence on one side of the ladder, you automatically know what's on the other side. It's a molecular backup system built right into the structure. Now, the order of these base pairs matters enormously. A sequence that reads ATGC means something completely different from TAGC. The complete DNA sequence in one human cell contains about 3 billion base pairs. 3 billion. If you typed it out 1 letter per second, it would take roughly 95 years of non-stop typing to finish. And this entire sequence is crammed into a nucleus so small you need a microscope to see it. If you stretched out all the DNA in one of your cells, it would be about 2 meters long. Now consider that this 2-meter string is packed into a nucleus only 6 micrometers across. That's like fitting a 2-meter thread into a sphere the size of a grain of pollen. DNA is carefully wound around proteins called histones, forming nucleosomes, which are then coiled and organized into chromosomes. Humans have 46 chromosomes, arranged in 23 pairs. 1 from each pair came from your mother, the other from your father. These chromosomes contain all your genes, the sequences of DNA that code for proteins, and proteins do essentially everything in your body. Some form structures like hair, others act as enzymes, still others fight infections or transport oxygen. Here's something you might not know. Only about 2% of your DNA actually codes for proteins. 2%. The rest was rather dismissively called junk DNA, which turned out to be spectacularly wrong. Much of it serves regulatory functions, controlling when genes are turned on or off. Some is structural, and some is still mysterious. The discovery of DNA's structure in 1953 is usually credited to James Watson and Francis Crick, but their work relied heavily on Rosalind Franklin's X-ray crystallography. Her photograph, Photo 51, showed the distinctive X-shape that revealed DNA's helical structure. Without her meticulous work, Watson and Crick would have been guessing in the dark. The model they built explained everything. How DNA could be copied, because you could unzip the ladder and each side could serve as a template. How information could be stored in the sequence of bases. How mutations could occur through copying errors. It was, in their published papers' language, of considerable biological interest. That might be the greatest understatement in scientific literature. Think about it for a moment. Every cell in your body contains the same DNA sequence, copied from the original fertilized egg you started as. That egg contained DNA copied from your parents, which was copied from their parents, back through an unbroken chain stretching back billions of years. Of course, the copying isn't perfect. Errors occur. Most are caught and corrected by molecular proofreading systems, but some slip through. These uncorrected errors are mutations, the raw material of evolution. Most do nothing. Some are harmful. Very occasionally one provides an advantage. You are, in a very real sense, a collection of successful mutations. Every difference between your DNA and your parents represents a tiny experiment in variation. Over millions of generations, these small changes accumulate into big differences. Different species, the astonishing diversity of life, all from modifications to the same basic 4-letter code. There's something rather beautiful about the simplicity of it, right? 4 letters. That's all life needed to create everything from bacteria to blue whales, from mushrooms to mathematicians. And right now, as you're drifting towards sleep, your cells are reading this code, following its instructions to replace proteins, repair damage, and keep you functioning. It's happening continuously, automatically, directed by sequences written billions of years ago and refined ever since. Now here's a question worth pondering. Where did DNA actually come from? Not from your parents, but in the deeper sense. Where did the very first DNA molecule come from on a young, lifeless planet? The answer is that we don't entirely know, but what we've figured out so far is genuinely fascinating and involves a surprising protagonist. Because DNA probably wasn't first. The current leading theory is that life began with DNA's slightly scruffier cousin, RNA. Ribonucleic acid is similar to DNA, made of bases attached to a sugar phosphate backbone, storing information in sequences. But there are crucial differences. RNA is usually single-stranded rather than double-stranded. It uses a slightly different sugar molecule, and it uses uracil instead of thymine. Still 4 letters, just with one substitution. Here's why this matters. RNA can do something DNA can't. It can fold up and act as an enzyme, catalyzing chemical reactions. DNA is brilliant at storing information, but it's chemically rather inert. RNA can be both the instruction manual and the machine that follows the instructions. This dual capability suggests something profound. If you were designing the simplest self-replicating system, RNA would be better than DNA. You could have an RNA molecule that contains instructions for copying itself and has the enzymatic ability to actually perform that copying. The RNA world hypothesis proposes that early life was based entirely on RNA. No DNA, no proteins, just RNA molecules that could store information, catalyze reactions, and replicate. DNA and proteins came later, evolutionary refinements that divided the labor. DNA became stable, long-term storage. Proteins became specialized workers. RNA became the messenger. So where and when did this begin? The best estimates place the origin of life around 3.8 to 4 billion years ago, shortly after Earth had cooled enough to have stable oceans. The planet was still rather inhospitable, frequent volcanic activity, no oxygen, intense UV radiation. But it had liquid water, and liquid water is chemistry's favourite playground. The question is, where exactly? Darwin himself suggested a warm little pond in 1871, imagining life beginning in a shallow pool rich in minerals, warmed by the sun, with lightning providing energy. It's romantic and might even be correct. The famous Miller-Urey experiment in 1952 showed that amino acids can form spontaneously under early Earth conditions. They sealed water and gases in a flask, zapped it with electrical sparks, and after a week found several amino acids had formed naturally. More recent experiments have shown you can form RNA nucleotides under plausible conditions, though we haven't yet produced a fully self-replicating RNA molecule in the lab. We're proving the building blocks can form naturally, but going from building blocks to a complete system remains one of biology's great puzzles. Another possibility is that life began near deep ocean hydrothermal vents. These underwater volcanic systems spew hot, mineral-rich water, creating chemical gradients and providing energy. The alkaline vents would have been ideal incubators, protected from UV radiation, stable, and continuously supplied with chemical energy. A third possibility is that life didn't originate on Earth at all. The panspermia hypothesis suggests that building blocks, or perhaps simple life itself, arrived on comets or meteorites. We know amino acids exist in meteorites, so it's not completely far-fetched. Of course, this just moves the question somewhere else. If life didn't start here, where did it start? Wherever it happened, the first self-replicating molecule marked a genuine threshold. Before that moment, Earth had chemistry. After, it had life. Not complex life, just a molecule that could make copies of itself using raw materials from the environment. And once you have something that can replicate, evolution begins. Copying is never perfect. Errors occur. Most produce non-functional molecules, but occasionally an error produces something better at replicating. That variant becomes more common. Over time, these early RNA molecules became more sophisticated, developing protective shells, learning to cooperate, eventually giving way to the DNA, RNA protein system all modern life uses. This transition probably happened fairly early. Even the most ancient bacteria used DNA. The RNA world, if it existed, was a brief experiment that left no direct descendants, only the genetic code it invented. There's something rather humbling about this, isn't it? That life might have started as an accident. A lucky combination of molecules replicating by chance, stumbling into complexity not through any plan, but simply because some combinations worked better than others. And here's the remarkable bit. That code has never stopped. From the first RNA molecule to you, there's an unbroken chain of successful replication. Every ancestor you've ever had managed to reproduce before dying. Not one failed. Not one broke the chain. You're the current endpoint of the longest-running success story in Earth's history. So we've established that DNA is ancient and remarkably good at copying itself. But here's where it gets personal, because the specific version sitting in your cells right now didn't appear randomly. You inherited it, half from your mother, half from your father, in a process that's simultaneously beautifully organized and utterly chaotic. You began as a single cell, a fertilized egg, when a sperm from your father met an egg from your mother. Each carried 23 chromosomes, half the usual number. When they fused, you ended up with the full 46, arranged in 23 pairs. But here's where it gets interesting. The specific chromosomes you inherited weren't simply copied wholesale. They were shuffled, mixed, recombined in a process that ensures you're genuinely unique. This shuffling happens during the formation of reproductive cells in a process called meiosis. Before your parents' cells divided to create the egg and sperm that would become you, their chromosomes paired up and physically swapped segments with each other. Picture two similar books, one from each grandparent. Before passing one to you, your mother cut sections out of each and swapped them, creating a hybrid that contains passages from both, but is identical to neither. This is exactly what happens to chromosomes. They line up, swap sections, and create new combinations that didn't exist in either parent. So the chromosome you inherited from your mother isn't purely your grandmother's or purely your grandfather's. It's a mosaic of both. You're not just a mix of your parents, You're a mix of all four grandparents, with a genuinely random selection of which bits you got. This is why siblings can look so remarkably different despite having the same parents. Each child gets a different shuffle of the genetic deck. You might have inherited your grandmother's nose and your grandfather's stubbornness, while your sister got completely different combinations. It's a lottery with 8 million possible combinations, and that's before accounting for the recombination swapping. There's something rather lovely and slightly melancholy about this. Go back enough generations and you'll have ancestors who contributed virtually nothing to your DNA. They existed, but through random shuffling, their genetic contribution got diluted to essentially zero. You carry no trace of them, though you might have inherited their uncomfortable Victorian furniture, which is arguably worse. But here's what you definitely inherited: mistakes. Quite a lot of them. Each human is born with about 60 to 70 new mutations that weren't present in either parent. Most are in non-coding regions and do nothing. But occasionally, a mutation does something significant. Most significant mutations are harmful, which makes sense. DNA is a finely tuned instruction manual refined over billions of years. Random changes are more likely to break something than improve it, rather like randomly changing words in a complex recipe. And yet, you're carrying quite a few broken genes. We all are. Scientists estimate each person has around 100 genetic variants associated with diseases, plus thousands more with subtle effects. Most don't cause problems because you have 2 copies of most genes, and usually 1 working copy is enough. But sometimes you inherit a broken version from both parents. When this happens, you might develop a genetic disease: cystic fibrosis, sickle cell anemia. These conditions are caused by inheriting 2 non-functional copies of a particular gene. It's a cruel lottery where both parents had to be carriers and you had to inherit the broken version from both. There's also evolutionary baggage cluttering up your genome. Take your inability to produce vitamin C. Most mammals can synthesize their own. Humans can't. We have the genes, but one is broken, disabled by mutations in our primate ancestors millions of years ago. Our ancestors ate fruit rich in vitamin C, so the ability to make it wasn't necessary. Now we're stuck getting it from our diet. Or consider your sinuses, excellent for infections, your appendix, which occasionally tries to kill you, wisdom teeth, which made sense when our ancestors had larger jaws, but now mostly just come in crooked and require surgical removal. These are examples of evolutionary compromise. Your body wasn't designed from scratch. It was cobbled together through millions of years of modifications, and sometimes good enough to reproduce was the bar. The result is a genome full of broken genes and design flaws that would make any engineer weep. And yet somehow it works. You work. Despite carrying broken genes, despite evolutionary baggage, despite billions of base pairs that need accurate copying, you're here, functional, listening to someone ramble about genetics. There's something rather comforting about this. You don't need to be perfect. Your DNA certainly isn't. It's a hodgepodge of ancient code, recent modifications, broken genes, and random mutations. And yet it's produced you, someone capable of contemplating your own genetic inheritance. You're not a perfect copy of anyone. You're a unique remix of everyone who came before, carrying forward the parts that worked well enough, along with all the quirks and flaws that didn't cause enough problems to be eliminated. Sleep well knowing you're exactly as flawed as you're meant to be. Right, let's talk about what DNA has actually achieved over the past few billion years. Because while we've been focusing on mechanics, we haven't addressed the bigger picture. DNA, through evolution, has created every living thing that's ever existed. That's rather an impressive CV. From bacteria to sequoias, from jellyfish to jaguars, everything alive is running on variations of the same basic genetic code. Same four-letter alphabet, same molecular machinery, just arranged differently and subject to billions of years of trial, error, and occasional spectacular success. Evolution by natural selection is breathtakingly simple. Organisms better at surviving and reproducing will, by definition, produce more offspring. Those offspring inherit the genetic variants that made their parents successful. Over generations, beneficial variants become more common while harmful ones disappear. There's no forward planning, no ultimate goal, just relentless, automatic filtering that keeps what works and discards what doesn't. And it's produced extraordinary solutions. Consider your eye. It contains roughly 2 million working parts, all working together to focus light and convert it into electrical signals. Darwin himself worried it seemed too complex to have evolved gradually. The earliest light-sensing organs were just patches of light-sensitive cells, barely able to distinguish bright from dark. Useful enough to be worth having, though. Over millions of years, these patches became more sophisticated. They developed a depression to determine light direction. The depression deepened into a cup. A transparent covering formed. A lens developed. We can trace this progression because different organisms today have eyes at different stages of complexity, from simple patches in flatworms to sophisticated camera-like eyes in vertebrates to bizarre compound eyes in insects. Evolution didn't set out to build an eye. It just kept whatever mutations made seeing slightly better. Or take flight. It's evolved independently at least 4 times. Insects, pterosaurs, birds, and bats. 4 completely different solutions, each using modifications of existing body parts. None were planned, they were accidents, random mutations that happened to be useful and got refined over millions of generations. DNA's greatest achievement is the sheer diversity of life it's produced. An estimated 8.7 million species currently alive, possibly more. Each one is a unique solution to survival, shaped by its environment and evolutionary history. Giraffes developed long necks. Cacti evolved spines and water storage. Arctic foxes grew thick white fur. Anglerfish developed luminescent lures. These aren't conscious adaptations. Random mutations occasionally produced variations, most doing nothing or actively harmful, but a tiny fraction providing some advantage. Those mutations spread. Over time, small changes accumulated into dramatic transformations. But evolution doesn't produce perfection. It produces good enough, which brings us to DNA's catastrophic failures. Let's start with cancer. Cheerful topic for bedtime. Cancer is fundamentally evolution happening inside your body. Your cells constantly divide, copying DNA, occasionally making mistakes. Most mistakes are caught. Some trigger self-destruct, but sometimes a mutation occurs in a gene-controlling cell division, and the cell loses its ability to regulate growth. That cell divides when it shouldn't. Its descendants inherit the mutation and divide as well. Another mutation might disable self-destruct. More mutations might allow invasion of other tissues. It's evolution doing exactly what it's supposed to, selecting for cells good at reproducing, except it's killing the organism those cells are part of. Rather a design flaw, a side effect of being made of cells that can divide and adapt. Then there are genetic diseases. Huntington's disease doesn't manifest until middle age, after most people have had children. Natural selection doesn't care what happens after you reproduce. Or sickle cell anemia, where inheriting 2 copies causes severe disease, but 1 copy provides malaria resistance. In regions where malaria is common, the gene persists despite harm when inherited from both parents. Evolution optimizes for survival and reproduction in your current environment, not for your health or happiness. If a gene helps you reproduce, it spreads, even if it causes suffering later. That's not cruel, just indifferent. DNA doesn't care about you personally, you're a temporary vehicle for your genes. Bit depressing when you put it like that, but there's another way to look at it. Yes, you're the product of billions of years of messy, imperfect evolution, but it's also produced consciousness, curiosity, creativity, and the ability to understand the process that created you. You can see both DNA's triumphs and failures, the sophisticated immune system and the overactive version causing allergies, the complex brain composing symphonies and the miswired version producing anxiety, the intricate sensory systems experiencing the world and the degenerative conditions sometimes taking them away. DNA has spent 3.5 billion years experimenting, keeping what worked well enough and discarding what didn't. The result isn't perfect, but perfection was never the goal. The goal is just to keep going, keep copying, keep trying variations. Judged by that metric, DNA has been spectacularly successful. You're carrying that successful code right now. It built you. It maintains you. And despite all its flaws and quirks, it's done a reasonably good job keeping you alive thus far. Not perfect, certainly, but good enough. For most of human history, DNA was rather like a book in a language nobody could read. We knew it existed, we knew it contained important information, but the actual contents remained mysterious. We could observe what it did, but we couldn't read the instruction manual itself. This changed dramatically in the late 20th century. Scientists developed techniques to sequence DNA, to read the precise order of those letters. At first, this was painstakingly slow. The first complete genome ever sequenced belonged to a virus with 5,386 base pairs, taking months. Your genome contains 3 billion base pairs. You can see the problem. But technology improved, and in 1990, scientists announced an absurdly ambitious project. They would sequence the entire human genome, all 3 billion base pairs, estimated cost $3 billion, timeline 15 years. The Human Genome Project was one of the most ambitious scientific endeavors in history. Hundreds of scientists across multiple countries worked simultaneously, each tackling different chromosomes, slowly piecing together humanity's genetic instruction manual. The project was completed in 2003, slightly ahead of schedule. Then scientists actually started reading what they'd sequenced, and some findings were rather surprising. First surprise, humans have far fewer genes than expected. Before sequencing, estimates range from 50,000 to 140,000 genes. The actual number? About 20,000 to 25,000. Only about twice as many as a fruit fly and fewer than rice. That's slightly humbling. This was genuinely unexpected. If genes code for proteins and proteins do all the work and humans are vastly more complex than fruit flies, shouldn't we have vastly more genes? The answer is that complexity comes from how genes are regulated, combined, and used. Human genes are more versatile, capable of producing multiple proteins through various splicing mechanisms. Quality over quantity. Second surprise, the vast majority of your genome doesn't code for proteins. Only about 2% consists of protein-coding genes. The rest, initially called junk DNA, turned out to be far more interesting. Some are regulatory sequences, controlling when, where, and how much of a gene gets expressed. These are crucial. Having a gene is one thing, but knowing when to turn it on and off is equally important. You don't want digestive enzymes in your brain or hair proteins in your liver. Regulatory sequences ensure genes are expressed in the right cells at the right times. Other non-coding regions are structural, helping organize chromosomes. Still others are evolutionary remnants, broken gene copies, fragments of ancient viral infections, repetitive sequences whose purpose remains unclear. Some we still don't fully understand. The third surprise, we're not as unique as we thought. Humans share about 99.9% of their DNA with each other. All visible variation comes from differences in just 0.1%. That's about 3 million base pairs, which sounds like a lot until you remember the total is 3 billion. But it gets better. We share about 96% with chimpanzees, about 90% with mice, about 85% with dogs, about 60% with bananas. Yes, bananas. You are, genetically speaking, more than halfway to being a banana. All life shares the same basic machinery because we descended from the same ancient ancestor. This molecular evidence provides some of the strongest support for evolution. You can trace relationships between species by comparing genomes. The more similar the DNA, the more recently they shared a common ancestor. Reading the genome also revealed our evolutionary history in unexpected ways. Our DNA is littered with remnants of ancient viral infections. About 8% of the human genome consists of sequences from retroviruses that infected our ancestors millions of years ago. These viral sequences are passed down now, fossil viruses preserved in genetic amber. Most are inactive, disabled by mutations, but some have been co-opted for useful purposes. One retroviral gene now plays a crucial role in the placenta, helping form the barrier between mother and fetus. Evolution is remarkably good at recycling. The genome also revealed something sobering: how similar we are to people with genetic diseases. Many disease-causing mutations are just single base pair changes. Change one A to a T, and you might disable a crucial protein. It's a reminder of how finely tuned our genetics are. Since the Human Genome Project finished, sequencing has continued improving absurdly fast. The first human genome cost $3 billion and took 13 years. Today, you can have yours sequenced for under $1,000 in about a day. We can now sequence cancer genomes, identifying specific mutations. We can sequence ancient DNA from Neanderthal fossils, discovering many of us carry Neanderthal DNA from ancient interbreeding. We can sequence thousands of individuals, studying genetic variation and identifying genes associated with traits or diseases. But reading the genome also revealed how much we still don't understand. Knowing the sequence is one thing, understanding what it means, how genes interact, how regulatory networks function, that's vastly more complicated, It's like having a complete symphony score but not knowing what most instruments sound like. We can identify genes associated with height, but there are hundreds, each contributing a tiny amount, and we still can't accurately predict someone's height from their genome alone. Same with intelligence, personality, susceptibility to common diseases. These traits are influenced by many genes plus environmental factors, creating complexity that defies simple genetic determinism. Reading the human genome was an extraordinary achievement, but it was also just the beginning. We've read the book. Now comes the harder part, actually understanding what we've read and deciding what to do with that knowledge, which brings us to the question of editing. So here's a question worth contemplating. If you could edit your own genome, would you? Remove a gene increasing your Alzheimer's risk, add one strengthening your bones, fix the mutation making you lactose intolerant. It's not science fiction. We can actually do this now. For most of history, deliberately changing DNA was purely theoretical. We could breed plants and animals, but that was slow and imprecise. We could use radiation to cause random mutations, which was like trying to fix a watch by hitting it with a hammer. Occasionally it worked, but mostly it just broke things. Then in the 1970s, scientists developed the first proper gene editing tools. They discovered enzymes that could cut DNA at specific sequences, allowing them to remove, insert, or modify genes. Revolutionary, but also clumsy. The enzymes weren't very precise. Success rates were low and the process was expensive and time-consuming. But in 2012, everything changed. Two scientists, Jennifer Doudna and Emmanuel Charpentier, published a paper describing CRISPR-Cas9. Calling it revolutionary is underselling it. CRISPR is to previous gene editing tools what word processors are to typewriters. CRISPR is actually borrowed from bacteria, who use it as a kind of immune system. Scientists repurposed this system to edit any DNA sequence they wanted. Here's how it works. You design a piece of guide RNA matching the DNA sequence you want to edit. This guide RNA attaches to a protein called Cas9, which acts as molecular scissors. The guide RNA leads Cas9 to the exact spot in the genome you're targeting. Cas9 cuts both DNA strands. Then the cell's own repair mechanisms fix the break, either disabling the gene or, if you've provided a template, inserting new DNA. It's elegantly simple, which is why it's been adopted so rapidly. Within years, thousands of laboratories worldwide were using it. The technique has been used to edit genes in bacteria, plants, animals, and yes, humans. Let's start with uncontroversial applications. Using CRISPR to modify bacteria or crops is relatively straightforward ethically. Scientists have created bacteria that produce insulin for diabetics, break down plastic waste, or generate biofuels. They've modified crops to be more nutritious, drought-resistant, or less reliant on pesticides. Editing animals is slightly more complex but broadly accepted for research and medicine. Scientists have created pigs whose organs might be suitable for transplanting into humans. They've edited mosquitoes to resist malaria parasites, potentially preventing millions of deaths. But editing human DNA? That's where things get genuinely complicated. There are two very different types with vastly different implications. The first is somatic cell editing, modifying DNA in specific body cells but not reproductive cells. If you used CRISPR to fix a mutation in someone's liver cells, that change would affect only that person and wouldn't be passed to children. This is already being tested in clinical trials for sickle cell anemia and certain cancers. It's essentially very precise medicine, treating disease by correcting the underlying genetic cause. There are risks, off-target effects where CRISPR cuts the wrong place, or incomplete editing where only some cells get modified, but these are technical challenges rather than fundamental ethical problems. Most agree that using gene editing to cure disease in living patients is acceptable, provided it's safe and effective. The second type is far more contentious. Germline editing involves modifying eggs, sperm or early embryos, changes that would be inherited by future generations. Edit an embryo's DNA, and that modification becomes permanent, passed down through that person's entire lineage. You're not just treating one individual, you're altering the human gene pool. In 2018, a Chinese scientist named He Jiankui announced he had created the world's first gene-edited babies, twin girls whose DNA he modified to make them resistant to HIV. The scientific community's response was swift and overwhelmingly negative. He had acted recklessly, editing embryos without adequate safety testing, without proper ethical oversight, and for a modification that wasn't medically necessary. Jiankui was subsequently sentenced to 3 years in prison, but it demonstrated something important. The technology exists, it works, and somebody was willing to use it. The question isn't whether we can edit human embryos. We can. The question is whether we should, and under what circumstances. Arguments in favour focus on preventing genetic diseases. If parents both carry a mutation for a fatal childhood disease, they currently face difficult choices. Germline editing could offer another option. Correct the mutation in embryos, ensuring healthy children who won't pass the disease to their offspring. Sounds reasonable, doesn't it? But here's where it gets thorny. If we accept editing to prevent serious diseases, what about less severe conditions? What about predispositions to diabetes or heart disease? What about traits that aren't diseases at all, like height, intelligence, or eye colour? There's a slippery slope. Once we start editing embryos for medical reasons, the technology exists to edit for enhancement. While nobody's seriously proposing designer babies yet, the technology that could prevent Huntington's disease is the same that could, theoretically, modify genes associated with intelligence or athleticism. Who decides what's legitimate medical use versus enhancement? Who has access to these technologies? If gene editing is expensive, does it become something only the wealthy can afford, creating a genetic divide? There's also the issue of consent. When you edit an embryo, you're making permanent changes to a person who can't possibly agree. Yes, parents make medical decisions for children all the time, but those are usually reversible or treat immediate problems. Gene editing is permanent and affects not just that child but all their descendants. And we still don't fully understand genetics. For most traits, especially complex ones like intelligence, dozens or hundreds of genes are involved, plus environmental factors. Editing one gene might have unexpected effects elsewhere. That said, there are genuinely compelling cases for germline editing. Some genetic diseases cause horrific suffering and early death. If we have the technology to prevent that, is it ethical not to use it? Would you, if you carried a mutation with a 50% chance of causing your child to die painfully in childhood, choose not to edit it if you could? These are uncomfortable questions with no easy answers. Most countries currently ban germline editing in humans, but technology tends to advance faster than regulation, and the tools are already widely available. So what do you think? If you could edit out your cancer risk, would you? What about for your children? Where would you draw the line between treatment and enhancement? It's worth pondering, because these aren't distant future scenarios. They're decisions society will need to make within your lifetime. We've reached an unprecedented moment in human history. For the first time, we have the ability to deliberately alter our own evolution, to rewrite the code that's been refined over billions of years. Whether that's a wonderful opportunity or a terrifying responsibility, rather, depends on your perspective. Probably both, if we're being honest. Right then, we've traced DNA from its murky origins in some ancient pool through billions of years of evolution, right up to the present moment where humans have learned to read and edit it. But where does all this go from here? What happens next in the story of DNA? And perhaps more interestingly, what happens to us? Let's start with the near future, the next few decades. The technology we've just discussed is improving rapidly. What's expensive and difficult now will likely be routine and affordable within a generation. Your grandchildren might live in a world where genetic diseases are simply edited out before birth, where cancers are treated by reprogramming immune cells, where aging itself is treated as a condition rather than an inevitability. That last one probably raised your eyebrows, didn't it? Aging as a disease? But there's serious research happening. Aging isn't just random deterioration. It's partly driven by genetic programs, cellular processes that evolved because they were beneficial early in life but become harmful later. Our cells have built-in limits on how many times they can divide. Our DNA accumulates damage over time. Some scientists think these processes could be slowed or even reversed. Extend telomeres, boost DNA repair mechanisms, clear out senescent cells. Whether this will actually work in humans remains to be seen, but the fact that we're seriously discussing it says something about how far we've come. But longer lifespans are just one possibility. Gene therapy is already treating previously untreatable diseases. Children with inherited blindness are regaining sight. People with sickle cell anemia are being cured by editing their own blood cells. These aren't science fiction. They're happening now in hospitals with real patients. Within decades, we might see treatments for Alzheimer's, Parkinson's, muscular dystrophy, conditions that currently have no cure. We might enhance immune systems to better fight cancer. We might repair genetic damage caused by radiation or toxins. The medicine of the future looks less like pills and surgery and more like precisely targeted genetic modifications. Then there's the rather extraordinary possibility of de-extinction, bringing back species that have gone extinct. Resurrecting dinosaurs isn't feasible because DNA degrades too quickly. But more recently extinct species? That's actually possible. Scientists have already sequenced the woolly mammoth genome from frozen specimens. The DNA is fragmentary, but enough survives to identify differences between mammoth and elephant genomes. You could, theoretically, edit elephant DNA to make it more mammoth-like, insert that into an elephant egg, and grow something resembling a mammoth. Whether this is a good idea is different. Where would these resurrected mammoths live? The Arctic ecosystem they evolved for no longer exists. Would they just be curiosities in zoos, or could they play a role in restoring tundra ecosystems? And should we be spending resources on bringing back the dead when we're struggling to save species currently going extinct? Similar projects are underway for other species. The passenger pigeon, extinct since 1914. the Thylacine, even the Neanderthal, though that raises even thornier ethical questions. Looking further ahead, perhaps 50 to 100 years, things get more speculative but no less fascinating. Some researchers envision humans deliberately modifying themselves for specific purposes. Enhanced immune systems for people working with infectious diseases, improved bone density for astronauts on long space missions, modified metabolism for extreme environments. This is sometimes called directed evolution, and it's fundamentally different from natural selection. Instead of random mutations being filtered by survival, we'd be making deliberate changes based on what we want to achieve. It's evolution with intention, guided by intelligence rather than blind environmental pressure. But here's the uncomfortable question. Once we start down that path, where do we stop? If we can edit genes to help astronauts survive in space, can we edit genes to make soldiers stronger? Can we create humans better suited for colonizing other planets? At what point does a genetically modified human stop being human and become something else? These aren't idle philosophical musings. They're questions we'll likely face within your lifetime, possibly within decades, and we're remarkably unprepared for them. Our ethics, our laws, our social structures are all based on a world where human nature was essentially fixed. That's no longer true. There's also the question of what happens naturally without deliberate intervention. Human evolution hasn't stopped. It's still happening, just in different ways. Modern medicine keeps people alive who would have died young in previous eras, allowing them to pass on genes that might have been eliminated by natural selection. Does this mean humans are getting weaker? Not necessarily. We've changed our environment so dramatically that selective pressures are completely different. We're no longer being selected for ability to hunt or avoid predators. We're being selected for ability to survive in modern environments with modern diets, medicines, and lifestyles. That's still evolution, just pointing in different directions. Some traits are definitely still being selected. Humans in populations that have farmed for thousands of years often have more copies of genes for digesting starch. People at high altitudes often have genetic adaptations for processing oxygen more efficiently. Evolution is ongoing, just subtle and slow compared to dramatic changes we can now make deliberately. Looking even further ahead, hundreds or thousands of years, things become genuinely speculative. Will humans still exist as we recognize them? Or will we have split into different varieties, some edited for Earth, others modified for space habitats or other planets? Could we diverge into separate species, unable to interbreed? It's possible. It's even likely if humans spread across the solar system and genetic drift acts on isolated populations. But it's also possible we'll remain one species, connected by travel and communication, sharing genetic modifications rather than diverging. There's also the possibility, rarely discussed but worth mentioning, that we'll simply stop being purely biological. If we can edit genes, we can also integrate technology, computer chips, artificial organs, direct brain-computer interfaces. The line between biology and technology is already blurring. Perhaps future humans will be hybrid entities, part evolved biology, part design technology, with DNA being just one component. Or perhaps none of this happens. Perhaps we decide the risks are too great, the ethical problems too intractable. Perhaps we choose to remain as we are, accepting our genetic inheritance with all its flaws. That's possible too, though I suspect unlikely. humans have never been particularly good at restraint when it comes to new technologies. What's certain is that DNA's story isn't finished. It's been writing itself for 3.5 billion years through blind mutation and selection. Now, for the first time, one species created by that process has learned to read the text and edit it. We're becoming co-authors of our own genetic future, whether we're ready for that responsibility or not. Where does that leave us, lying here in the dark contemplating our genetic code? I think it leaves us at a rather extraordinary moment. We're among the first humans to truly understand where we came from, to read the ancient text in our cells, and to glimpse where we might be going. You're carrying a code older than mountains, refined by billions of years of evolution, and now on the cusp of deliberate modification. You're part of an unbroken chain stretching back to the first self-replicating molecules, and potentially forward to futures we can barely imagine. That's worth pondering as you drift off. You're not just a person, you're a chapter in the longest story ever told, written in 4 letters, billions of years long, and still being written. And that brings us to the end of our journey through the story of DNA. From mysterious molecules in ancient pools to the sophisticated code that built you, from blind evolution to deliberate editing, from the deep past to an uncertain but genuinely fascinating future, it's been quite a journey, hasn't it? If you're still awake, which honestly would be a bit disappointing given the entire point of this exercise, I hope you've at least learned something from this journey. But for now, for this moment, it's enough to simply exist. To lie here, breathing slowly, feeling your body doing all the unconscious things that DNA programmed it to do. Your heart beating, your lungs expanding, your brain gradually beginning to switch off for the night. If you've enjoyed this gentle exploration through genetics, a like would be tremendously appreciated, and if you'd like more of these calm journeys through science, do consider subscribing. It genuinely helps this little channel grow and lets me know you'd like more bedtime science stories. For those of you who've been here before, who keep coming back to these quiet moments, thank you. Your presence here means more than you know. I genuinely enjoy making these videos, and I enjoy even more that you're here to listen to them. For anyone still awake, I'll leave you now with some soft ambient sounds to help you drift off properly. If you've already fallen asleep, well, it means I did my job right. I hope you have pleasant dreams of double helixes and evolutionary trees, of ancient molecules and future possibilities, of a code that's been running for billions of years and shows no signs of stopping. Until next time, sleep well and good night.